Hi everyone, I'm Mrs. DeForce. Join me today in unlocking the secrets of life. Today we embark on a journey into the fascinating world of bacteria, the unsung heroes and notorious villains of the microcosm. Bacteria are single-celled microorganisms found almost everywhere on Earth. They inhabit diverse environments from deep sea vents to your kitchen sponge. So let's take a look at them. Bacteria are classified as prokaryotic organisms. Prokaryotes are a group of organisms that lack a true cell nucleus with membrane-bound organelles. So the term prokaryote comes from the Greek word pro, meaning before, and karyon, meaning nucleus, or together we can say it means before a nucleus. This characteristic sets up prokaryotic cells apart from eukaryotic cells. So bacteria are a large and diverse group of microorganisms that belong to the domain bacteria. They're classified as prokaryotes because their cells lack a membrane-bound nucleus. In other words, they do have genetic material, but the genetic material is not surrounded by a membrane. They also have areas within the cell that does the functions, for example, of cellular respiration, but they don't have a typical mitochondria that is surrounded by a membrane or an endoplasmic reticulum, which are found in all eukaryotic cells. Now, bacteria come in various shapes, and their morphology is one of their essential characteristics. So we can identify some spherical shaped bacteria, rod shaped or cylindrical shaped bacteria. They are usually called the bacilli. We have spiral shaped bacteria, which is usually called spirilla. Comma shaped bacteria. We have corkscrew shaped bacteria. So the shapes are quite various. Bacteria can then arrange themselves in different ways based on their shape and cell division characteristics. The arrangement of bacterial cells can provide insight into their growth and division patterns. So here are some of their common arrangements. First of all, we have the cocci. The diplococci is cocci that is arranged themselves into pairs, often following cell division. So directly after cell division, you find that there are two of them together. Let's see, they are double, diplo. Tetrads are also found when they arrange themselves in groups of four or clusters. Micrococcus would be a typical example of this. Then you have the cocci that arranges themselves in cubical packets. In other words, it's clusters of 8 cells together. This would be an example like Sarkinia. They are relatively rare in medical microbiology. When we're looking at the bacillus shapes, first of all, we can get those that have just the single bacillus. Then we have those that are arranged as a double bacillus called diplobacilli. So the bacilli arrange themselves in pairs, again, directly after cell division. Then we have strings of bacilli, like here, we have strings of bacilli. They form chains because as they just divide, they form these long chains. Then we also get the palisade arrangement where the bacilli align themselves side by side like a row of fence posts. This arrangement is seen in some species of Corynebacteria. Then we have your spiral or helical bacteria. The spiral or helical shaped bacteria have very rigid structures, and examples include Spirillum campylobacter species, and the spirochetes are spiral bacteria with a very flexible corkscrew-like shape. Examples here would include Treponema, the causative agent of syphilis. The arrangement of bacterial cells is influenced by several factors, including their shape, their division planes. In other words, do they divide from the tip or do they divide on the side? And the presence of cell wall features, specific features within the cell walls. These arrangements can be useful in lab conditions for identifying bacterial species and understanding their growth patterns. Keep in mind that bacterial arrangements can vary even within the same species under different growth conditions or stages of growth. Additionally, certain arrangements can be indicative of species of specific bacterial pathogens, which can aid in diagnosis and treatment. If we look at the basic structure of bacteria, bacteria have a relatively simple structure compared to eukaryotic cells. As part of their structure, they are usually unicellular. In other words, a single cell can survive on its own. So it can be said that they are single-celled organisms or unicelled organisms. The genetic material, as mentioned, they are prokaryotic organisms. And then there's a very interesting genetic material called the plasmid, which is circular DNA found inside the bacterial cells. Now, this is a very important feature in biotechnology. If you want to, you can look at the video based on bacterial genetics, and it's used in biotechnology. And then we have different kinds of organelles. Previously, I mentioned that they don't have a specific mitochondria, but they do have cellular respiration. This function is done by the mesosome. And then some bacteria, but not all bacteria, will also contain chlorophyll, but they don't have chloroplasts. And then the cell wall is a very, very interesting feature of bacteria. The cell wall is often used in the classification of bacteria. We can distinguish between gram-positive and gram-negative bacteria. The primary difference between them lies in the composition and characteristics. The cell wall composition of a gram-positive bacteria have a very thick peptidoglycan layer. This whole layer here is the peptidoglycan layer in their cell walls, which makes up a significant portion of the cell wall structure. Peptidoglycan is a polymer made of sugars and amino acids. In gram-positives, the peptidoglycan layer is thick and forms a multi-layered structure, making the cell wall relatively rigid and robust. Gram-positive bacteria also contains teichoic acids embedded within the peptidoglycan layer. These compounds play a role in the cell wall stability and may contribute to pathogenicity. The gram-positive bacteria lack an outer membrane outside of this peptidoglycan layer. So in the gram staining procedure, gram-positive bacteria will retain the crystal violet stain due to their thick layer, which traps the dye. If we then look at the gram-negative bacteria have a very thin peptidoglycan layer. It's literally just a very, very, very thin layer in their cell walls. In gram-negatives, the peptidoglycan layer is sandwiched between an inner cytoplasmic membrane and an outer membrane, making it less prominent. In gram-negative bacteria, the outer membrane is composed of lipopolysaccharides. We abbreviate them as LPS. They also contain phospholipids and proteins. This outer membrane, this outer part of it, provides an additional layer of protection. The space between the inner and outer membranes in gram-negatives is known as the periplasmic space, and it contains the enzymes and proteins involved in various cellular processes. During the gram staining procedure, gram-negative bacteria do not retain the crystal violet stain due to the thin peptidoglycan layer. Instead, they will take up the counter stain safranin and turn pink using this staining procedure. So in summary, the key difference in the cell wall structure between gram-positive and gram-negative bacteria is the thickness and complexity of the peptidoglycan layer and the presence of an outer membrane in negatives. These structural differences are important for bacterial classification, antibiotic susceptibility, and the development of treatment strategies. Later, we will look at antibiotics and how bacteria can become antibiotic resistant. I'll put a link down in the description as well. Bacteria exhibit various modes of nutrition. Bacteria that produce their food through processes like photosynthesis or chemosynthesis is known as autotrophic bacteria, while bacteria that obtain their nutrients from external sources, including organic matter, is known as heterotrophic bacteria. Another feature is movement. So some bacteria can move using a structure like the flagella. Some of them will have one, some of them two, some of them will be surrounded by flagella, or they can have gliding motions for movement. Some bacteria are stationary, so wherever they land, they attach to the surface and there they will stay. Another important feature is the fact that bacteria can reproduce very rapidly through a process called binary fission. Binary fission is basically mitosis that takes place. A cell makes an identical copy of itself. And in bacteria, this process is very, very quick, sometimes up to less than 20 minutes to divide one cell into two identical daughter cells. Now, this is a very important characteristic of bacteria that makes them ideal in the use of biotechnology. Again, later on, if you click down in the links below, you will find access to a video that explains the importance of bacteria in genetic engineering and in biotechnology. Bacteria in this form can then grow exponentially. So exponential growth will be described in our chapter on population dynamics. If you want to click on that, you will find some interesting information on bacteria and how they grow in nature. Bacteria's adaptability is a hallmark of their success. Bacteria are extremely diverse genetically, and they have the ability to transfer their genes horizontally in a bacterial community between different species of bacteria. Bacteria occupy diverse habitats from extreme environments to just in our bodies. And bacteria often play vital roles in our lives. Gut bacteria, for example, aid in the digestion and nutrient absorption. Bacteria can also be used in biotechnology for insulin and antibiotic production. I will have a link below so that you can go back and check the video on bacteria and their use in genetic engineering and biotechnology, as well as there's another one on the development of antibiotic resistance in bacteria as a form of natural selection. Bacteria also help break down organic matter in ecosystems. Let's look at a few examples. Staphylococcus is a genus of bacteria commonly found on the skin and mucous membranes of humans and animals. They are gram-positive spherical bacteria that often occur in clusters resembling grapes, which is where their name, Staphylo, meaning clustered, and coccus, meaning spherical, originates. Staphylococcus bacteria are widespread in the environment, and they are part of the normal flora of the skin and mucous membranes of both humans and animals. Another interesting one is Lactobacillus. Lactobacillus bacteria are typically rod-shaped, known as bacilli. They're commonly found in the human gastrointestinal tract and are involved in the fermentation of sugars into lactic acid. Lactobacillus is used in the production of various dairy products like yogurt and cheese and is considered beneficial for gut health due to its probiotic properties. But some bacteria can also cause diseases in humans, animals, and plants. So if we look at an example, Heliobacter. Heliobacter is the most well-known species in this genus. It is helical or spiral-shaped. Heliobacter pylori is known to inhabit the human stomach and is associated with the development of gastritis, peptic ulcers, and some types of stomach cancer. Research on this bacterium has significantly contributed to our understanding of the link between bacteria and disease. And another example is syphilis. Syphilis is not necessarily a bacterium, but a sexually transmitted infection. It's caused by, however, the spirochete bacterium called Treponema pallidum. This has a distinctive spiral or cork-shaped structure. Syphilis progresses through several stages, starting with sores at the infection site and potentially leading to severe systemic effects if left untreated. Please get to a doctor. Want to dive deeper into life sciences? Check out more of our videos. Don't stop learning now. Explore more videos and download our worksheets for a complete learning experience. Links will be found below.